Have you heard what's happening in Calgary, Canada? Home to some of the world's best researchers and innovators in life sciences, Calgary is advancing healthcare solutions to solve global challenges. Calgary's dedication to the life sciences sector is evident in its labs, hospitals, schools, and the minds of its people, with its top institutions producing internationally recognized research and more than 110 life science companies backed by a highly skilled pool of talent. The life sciences sector is accelerating innovation in Calgary. If you're a bright mind or a bright company, Calgary is just the place for you. Take a closer look at calgarylifesciences.com. I'm Daniel Levine, and this is the Bio Report. As concerns grow about the use of animals to test drugs, scientists have sought new ways to analyze the efficacy and toxicity of their products. Genoskin is seeking to enable better, faster, and safer drug development through the use of its proprietary ex vivo human skin platforms for preclinical drug testing. The company provides natural human skin obtained from patients that it maintains in a living and functional state. We spoke to Pascal Descargues, founder and CEO of Genoskin, about the company's ex vivo human skin models, how they provide an alternative to animal experiments, and how they can accelerate the generation of reliable human data in drug development. Pascal, thanks for joining us.
Thank you, Danny, for gently inviting me.
We're going to talk about Genoskin, how you're able to provide human skin for preclinical testing, and how this can reduce the need for animal testing. Before we get into Genoskin's offerings, there have been pressures to move away from animal testing. How much of this is driven by ethics, and how much is this driven by utility? How predictive are animal models of drugs in humans?
Yes, this is a very interesting question, and so there are two factors. So ethics first, and also I would say the science and all experience, data, knowledge we have now about, for example, a drug development. And probably you may know that 90% of drug development fails during clinical trials. And this is something it's fantastic how people can accept this. This causes a loss of more than 1 billion per drug. If you estimate that the drug development, the total cost is about 1 to 2 billions, 10 to 15 years of work, and at the end, you have 9 projects that will fail among tens. And I think it's really a failure that needs to be overcome, and I'm not sure everybody should accept this. So, and we know that today, one reason is because of the poor predictability of animal models used during preclinical tests. So you validate drugs in animals that are not necessarily mimicking the human situation. And when you do a clinical trials, you have this failure because of loss of efficacy, because of toxicity in human. And so you stop everything, all these 10 to 15 years process and all these billions or millions dollars of investment. So for us today, for Genoskin, I think that really what is driving our growth, what is driving the possibility to provide alternative to animals use, it's more and more based on science, because we need something to de-risk drug development. And I can mention also the ethic issue here, because obviously there are more and more people in the world that wants to protect animals, and you have all this story with the meat, the vegetal meat, the alternative to the meat, the alternative to animal testing for cosmetic. It's true in Europe. It's true in many other countries. It's true now in California, here in the US. So there is a pressure, but today this is mostly related to non-therapeutic products, I would say, or cosmetic, or some chemicals, and not necessarily drugs. And finally, it makes sense. You want to develop cosmetic actives, and it's not a drug. It's something for your own pleasure, for example. But people will not accept now that we test everything on animal. We test the active on the animals. But for a drug, you have patients that are suffering. You have people that are dying. So the needs are different, and much more based on the science. And what do you want? You want to lower the attrition rate, accelerate the drug development, and at the end provide more and more reliable drugs for patients.
You use actual human skin. How do you obtain this?
So we use actual skin. Actually, we work closely with hospitals to ethically source these human skin leftovers. They come from plastic surgeries, and we ask for the consent from the patient before sourcing, before collecting these skin samples. And I think it's really the philosophy of Genoskin. You have a lot of companies that are spending millions to develop artificial tissues to try to replicate organs, human tissues. At Genoskin, we wanted to try an alternative to this pathway because there are more and more tissues available in the world. For example, for the skin, only in the US, every year, you have 200,000 skin samples produced from abdominoplasty surgery. It means that the vast majority of these skin samples are discarded. Actually, hospital and clinician, they pay to destroy these particular biological samples. And for us, we think that these tissues are actually the perfect matrix to develop tools, in vitro tools, for research instead of discarding everything. So it's how we ended in this particular field. But to do so, we need to have IRB approval. We need to comply to a lot of ethical and legal requirements. And of course, because it's coming from a person, a living human subject somewhere here around you, we need to ask for the consent. So everything needs to be very ethic.
And is something done to keep the tissue alive or prepare it for further experimentation?
Yes, and I think so. The skin, we did not invented the skin because it's from plastic surgery. So we just use this advantage. Um, but what we did or we are providing to our customer today, it's a proprietary um, nourishing gel. So we use a nourishing gel to keep run skin biopsies about 2 inches um, alive, functional, and importantly, immunocompetent for 7 days. So when we receive the tissue, we process the skin very rapidly in our facility, and we produce this skin model, ex vivo human skin models, that are embedded in this nourishing gel matrix. And we maintain this tissue alive for a couple of days, up to 7 days, so we can test drugs. We can also deliver them as a ready-to-use kit to our customers.
My understanding is that Genoskin is not the only company to provide skin on the market, but how does your offerings differ from what else is available?
Yeah, of course, we are not alone, and I think it means that the needs are very important. And today we have two main competitors. We have all these companies that are bioprinting or reconstructing tissue or skin. And the main limitation, as I mentioned today, it's artificial in the sense that it's made by the human, made in the lab with different cells put in 3D. And it's not true that you can replicate a complex organ like human skin in a lab. It's not accurate. So there are a lot of limitations. The tissue is not mature. It's not functional like adult skin, for example. And other competitors are finally biobanks. And this is all the companies. Some of them are very old companies. They are sourcing the tissue. Usually they keep the tissue at minus 20 frozen for a while, and then they provide some piece of skin or tissue to customers. So the main limitation here is that when you freeze or you put at 4 degrees, it's the same. You will kill the tissue. A human tissue or the skin organ is made to be maintained at 37 degrees. Not frozen or not put in the fridge. So this is the main limitation. And what we provide that is really unique is that we provide either a ready-to-use kit. So it means it's no longer a human skin sample. It's a kit made of standardized, ready-to-use skin models placed in multi-weld plates with and together with a culture medium that has been validated. So our customers can order and we can deliver them for reliable testing at their facility. Or we can also leverage in our lab to perform more advanced testing of their product. So the difference is in the way to keep the tissue alive, functional, immunocompetent, and even more injectable. With our system, we have the first platform to inject in human, outside of a human.
So how three-dimensional are these samples, and how well do they imitate human skin in vivo?
So I can mention, so the diameter usually is about 2 inches, and thickness is about 1 inch. So it's really it's not a small teeny piece of 3D cells. It's a big piece of tissue, and thanks to the our proprietary nourishing gel, we actually maintain a tissue tension, the tension on the skin, like in vivo, like with your own skin. And also we keep the skin surface in contact with the air, the atmosphere. Only the underlying tissues like the dermis or like the hypodermis are embedded in this nourishing gel matrix and are in contact with the culture medium we provide. So we mimic this interface between air, the atmosphere, and the tissue below, nourished by the culture medium, like the blood, for example, in our body. So this is very similar to your skin.
You actually have different offerings. I'm wondering if
Yes.
you could walk us through these. You have the native skin, the hyposkin, and the inflammaskin. What are each of those, and how do they differ? How are they used?
So, yes, and the first product we launch, it's native skin, and it's made of epidermis and dermis, and it's really dedicated to test topical products or formulations or drugs. With native skin, you can apply a cream, you can perform massages, and then you can test the efficacy or the toxicity of the product into the skin. Hyposkin, it's a newer technology. And the only difference, it's very simple. We have in the hyposkin the fat tissue plus the epidermis and the dermis. And the total thickness is about 1 inch. And with the fat tissue, with this called the hypodermis, we can use a syringe and a needle to inject in our hyposkin platform a drug, a vaccine, monoclonal antibodies, insulin, all these drugs that are developed today for subcutaneous injections. So, and this is totally unique. It's the first time a company provide a technology, a platform to test skin injection outside of a human, but in a human organ, not in an animal. So this is the uniqueness of this offer, and in particular with hyposkin. And recently we mentioned this in a press release. We can check what we call injection site reactions. So injection site reactions, how local adverse events like erythema, swelling, and pain that are commonly reported after vaccine or biologic injections. I don't know if you got your vaccine, your anti-COVID vaccine injection, but perhaps you had some pain after the injection, and probably it was a little bit red on the skin, and this is called injection site reactions. And today, you don't have any animals models to really test these particular injection site reactions. So how technology hyposkin enable this, we can better understand how a product induce these particular local adverse events. And so finally, the last technology we have, it's called inflammaskin. And here it's a little bit different. It's a psoriasis-like skin model. We use normal human skin. We treat the skin to become a psoriasis skin-like. with an inflammation, Th1, Th17 inflammation, and the inflammatory skin models enable the testing the efficacy of anti-inflammatory drugs, either topical drugs like steroids or skin injected drugs like Humira, for example, or other monoclonal antibodies.
How consistent are results from one sample to another? How reproducible are experiments conducted with Genoskin's products?
So for reproducibility, we are working on how the skin models are prepared, how they are maintained, how we are administrating the drug by injection or not. And we try to make sure everything is reproducible, especially we are developing today robots, where we are using some robots to help us to be more reproducible. And regarding the skin itself, obviously there are variation among donors. One donor will not have exactly the same skin or the same gene expression, the same immunological system or signature in the skin than another donor. And this is, instead of having a limitation here, we think it could be an advantage because we can source a lot of skin samples from different donors very rapidly. We actually receive skin samples every week. We can reproduce almost small clinical testing with different donors. If you want to test on male and female, and some donors age of 30 and others of 60, we have this. We have a little bit this diversity, human diversity in our samples, and it's how we can provide more accurate data for our customers, because we provide like first in human data, and we can test their product in 5, 10, 15, 20 different donors. So it's really a different way and compared to other platforms of cells, for example, that are more involved in drug discovery, for example.
And what's the business model? Do you operate as like a CRO, or do you just provide these tissue samples for drug developers?
We are doing the two offers. We are providing these two offers. So we are what we call a partner research organization, because we are not necessarily only doing the work a company would not have the time to do. We provide a unique platform. We provide all the expertise. We provide all the tools, for example, artificial intelligence-based tools to analyze the results and everything. So this is the service part of our activity, and the other part is to provide the skin models in ready-to-use kits. We deliver them everywhere in the US. We deliver them in Europe. We deliver them to Asia. And it's not necessarily the same kind of customer that will order the skin models, or that will order a particular service study with us. So we are like doing both today.
And are there any regulatory hurdles for you? Have you done anything to validate this, or had discussions with FDA?
So we do have. We did a lot of work to validate our technology, and actually we are publishing. We have some articles. we want to publish more. We work also with either customers or key opinion leaders to publish this data. But today we don't need a particular approval for our product, because our skin models are not necessarily used for the regulatory toxicity testing of cosmetic products. It's more for efficacy, and efficacy is not regulated. And regarding the drugs, we actually went almost 2 years ago at the FDA. We were invited by some people at the FDA to better explain them how our platform can be used to validate, for example, toxicology in the skin or immunotoxicology of drugs or subcutaneous injected drugs, for example. So they are aware of this, and I think they are also looking for these new technologies, like how technologies to bloom and be available for drug makers to better validate, better secure the drug development. So we don't have a particular approval, but everything rely on the quality of what we are providing, and also the pertinence of the scientific approach behind our platform.
Pascal Descargues, founder and CEO of Genoskin. Pascal, thanks so much for your time today.
Danny, thank you so much for your time.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.